anyone can generate really massive amounts of next generation sequencing data now, as many of you are aware. The challenge is often analyzing it. And we can talk about, filtering. Gordon always likes to talk about, Gordon's obsessed with filtering. I'm not convinced that it's, I don't like to emphasize this filtering idea as much as Gordon does, but basically, no matter how much filtering you do, there can always be issues that come up that you never would have anticipated. And whether we, sometimes we forget, but some of the studies we do and the things we say and write can really have profound implications and can cause a lot of actions to be taken. And so, I think it's worth being really, it's worth being careful. And this is the story. It's about Flathead Lake, basically. It's about the Flathead Basin. It's about bull trout that are threatened in the Flathead Basin. And I guess I don't want to give away the punchline, but basically we decided to turn this into a paper titled Caution and Skepticism Are Critical When Analyzing Massively Parallel Sequencing Data, so basically NGS data. And I'm going to sort of tell it how it happened from our perspective, okay? And yeah, so feel free to ask questions during it. We got all sorts of time also, I'll say. So this was a project led by my former master's student, Zach Norgard. See if we can figure that out. Okay. So basically, so these bull trout are really amazing fish. I think you guys will learn about them tomorrow. Gordon, will Clint be talking about bull trout tomorrow on the boat rides?
Yeah, on the boat rides will be discussing lake trout, bull trout controversy.
Yeah, so these really interesting fish that live throughout the Flathead Basin that have really declined severely over the past several decades. And basically, Gordon is involved, and other people around here are involved in another number of projects to monitor bull trout, estimate effective population size, characterize population structure, these types of things. So inevitably, we got involved in some of the projects. We collaborated a lot with Gordon, and we generated some rad data to discover SNPs, and we were going to use these data for various things. But basically, one of the first things we wanted to do after we generated a bunch of data was we basically wanted to investigate the relative health of these 6 bull trout populations that were collected throughout the Flathead Basin. So here's where we're at. This is Flathead Lake. We're right in here somewhere, and these are 6 populations that are collected upstream. And basically, what I really like about, one of the things I really like about population genetics, and many of you guys are aware of this, is that anything that happens in a population influences the patterns of genetic variation in that population, whether it's demographic events or selection, adaptation, these types of things. These things influence patterns of variation. So we thought, well, let's explore patterns of genetic variation in these populations and see if we can figure out some things that have been going on in the populations. Basically, try to assess their relative health. And so we generated a bunch of rad data from these 6 populations. And the first, we typically discovered tens of thousands of SNPs, and the first thing we wanted to do was just look at population structure, right? So we called all these genotypes just using our standard methods, and basically we did a PCA to look at population structure. And we saw a strong structure. Each of these individual locations, these individual populations was a distinct group. You're only looking at 2 axes of the PCA. These resolve in here if you look at additional axes. But it looked like what we'd expect. One thing we noticed was that the most divergent population was this Quartz Lake population. So this is Quartz Lake is a lake that's on a tributary of the North Fork of the Flathead. And one thing we noticed too right away was there were these 3 individuals within Quartz Lake that seemed to be more distinct. We didn't really think much of it at the time, but we thought, Quartz seems to be this interesting population. So the next thing we did was we decided, well, let's look at what's called the site frequency spectrum of these 6 different populations. Okay, basically, if you take coalescent theory and a Wright-Fisher model, you can convince yourself that a neutral population should have a particular spectrum of allele frequencies. Okay, without going into theory behind that, but basically, this is what a allele frequency, a derived allele frequency spectrum looks like for one of the populations. So what you're looking at on the x-axis is basically the derived allele frequency. So every time you have a biallelic SNP, one of those alleles is the ancestral allele and one of them is the derived allele. And so if you have 10,000 SNPs, you can basically, for each allele, you can quantify, or for each site, you can quantify the derived allele frequency, and you can bin those and basically make a histogram. So there's some proportion of polymorphic sites that have a low derived allele frequency, higher and higher and higher. Okay? So a Wright-Fisher model, coalescent theory, basically predicts a derived allele frequency that's steeper than this. Okay, so this say frequency spectrum was basically flatter than what we would expect if the population was constant size. So this is consistent with a population that would have gone through a relatively strong bottleneck. Okay, so Whale Creek. We looked at another population, Nonian Creek, and it was also the same thing. So the site frequency spectrum was much flatter than what would be expected if the population was maintaining a constant size. Okay? So it seemed like all the populations we looked at had this site frequency spectrum that was indicative of a strong and recent bottleneck. Basically, our plans, this was just sort of, we were just looking at the data, exploring things. You can take these site frequency spectra and you can model the strength and the timing of bottlenecks and these types of things. But this was sort of exploratory at this point. We were just looking at some of these allele frequency spectra of the different populations. And so, but this one population, Quartz Lake, had a site frequency spectrum that looked totally different. Okay? So instead of being flat, and let me just say this too, this is important note. So what I said here is what we do is we infer the ancestral and the derived state of each polymorphic site. And the way that we do that is we take reeds from a different species, from an outgroup species. In this case, we use lake trout. We align them, and we basically use lake trout to polarize or to infer the ancestral state of all the different polymorphic alleles. Okay? So we had some lake trout data sitting around as well. We aligned the lake trout reeds, and we basically inferred ancestral state using lake trout. Okay? And so when we looked at this next population in Quartz Lake, it had a really crazy looking site frequency spectrum, right? So this is like this half pipe thing where it had a really high frequency of sites that had a high derived allele frequency. So normally, a site frequency spectrum should look like this. This is a pattern that sometimes you see if there's a selective sweep at a particular locus, but you don't ever expect to see a genome-wide pattern like this. So a genome-wide site frequency spectrum that looks like a half pipe like this. And so first, I was convinced that my student Zach had just, I don't know, done something wrong with the bioinformatics or whatever, right? You can run all this stuff and you get all sorts of crazy things. And that happens to us too. And I thought, okay, you did, I don't know what was going on, just rerun it and check everything. But he kept running it and was checking everything, and we just kept getting an allele frequency spectrum that looked like this. And none of the other populations looked like that. And so we started to think, well, I guess this is real, but what on earth could have caused an allele frequency spectrum to look like this? And so basically, we started thinking about this, and I thought, the only thing I could think of, does anybody have any idea what could cause this? Jeff, what about you? Any idea?
Yeah, I talked to Gordon about it before, but if it was biological, it would be massive integration.
Yeah, exactly. So we basically, the only thing we had thought of was, man, it could be hybridization. Somehow, basically these bull trout, if they're hybridizing with lake trout in this one location, there's a bunch of lake trout alleles at a low frequency, basically, and these lake trout alleles at a low frequency in this population would give us a site frequency spectrum that looks like this. Okay? Now, no one had reported hybridization between bull trout and lake trout before, but we started talking to, and that would be just a disaster if these bull trout and lake trout were hybridizing. And so we started talking to Clint Woolfield, who you'll meet tomorrow. And first he said, no way, there's not hybridization between bull trout and lake trout. But we said, well, it's a really strong signal in the data. And he said, well, is there any way that there was contamination? And we said, well, no, it could not possibly have been contamination because we had prepared the bull trout samples in my lab before we had ever had lake trout in the lab. So there was no way that lake trout DNA could have contaminated the bull trout samples. We just had never had lake trout DNA in the lab, okay? And then I was telling him this, he said, well, what's interesting about Quartz Lake is that it's a really lake-like bull trout population. So these bull trout stay in the lake and they migrate up this little tributary to spawn. They're not thought to migrate in the same way that many of these bull trout populations migrate down to Flathead Lake. And one of the things that Clint said was, on some years, especially years when it's really dry, there's very little snowpack, the creek that feeds into Quartz Lake is really small. And he could imagine that there are particular years where bull trout have trouble actually getting up the creek to spawn. And he said, when we gill net these lake trout, so there's big projects going to remove lake trout using gill netting in various lakes around here. And when they gill net these lake trout, he said, sometimes we pull up ripe bull trout, mature bull trout, on the spawning beds where we're gill netting the lake trout. So even though bull trout, are supposed to spawn in creeks or rivers, and lake trout are supposed to spawn in lakes, it seems like this is a situation where there may be bull trout spawning with lake trout, and especially on dry years with little snowpack. And then we started thinking, oh man, with climate change, there's going to be less snowpack. There's going to be this, increased hybridization between these species. And so, we were really thinking about this, but we said, well, we want to be really sure that this is going on. But we discussed this and we really thought, wow, this seems like a situation where we've detected these novel hybrids. And it was very concerning. And so what we thought was, the first question was, we saw this spectrum, and I guess I should have showed you a little more data before I told you all that story, but basically we said, okay, I wonder if this is going on, if this could be caused by hybridization. So we thought the easiest thing we could do is we'll just do a PCA, but now we'll include some lake trout samples in addition to the bull trout samples that we had, okay? So we just did a PCA, and this time we included a bunch of lake trout as well as all the bull trout. And what we saw is that these 3 bull trout that we had seen before, that were these 3 bull trout in Quartz Lake, were basically pulled in the direction of lake trout on this PCA analysis, okay? So at this point, and that was actually what we sort of had expected, because the only thing we could think about was a low frequency of of lake trout alleles in that population to cause this site frequency spectrum. So basically, we said, well, what is going on here? Again, we knew that it wasn't, these same 3 bull trout samples showed up as the sort of putative hybrids, okay? So we thought, well, how is this happening? We need to be really sure. If these are really hybrids, we need to convince ourselves that this is not somehow a mistake in the bioinformatics or, contamination. We knew it wasn't contamination, but we sort of really wanted to try to be thorough and test this extensively. So what we did was we said, well, let's instead of doing a PCA like this, let's just simplify it, and let's try to define species diagnostic SNPs, okay? So we had many, many lake trout samples from throughout the range, and we had, a couple hundred, hundreds of, or 100 or so lake trout samples, I believe, 100 or so bull trout samples. They were each, the bull trout were sampled, from throughout the Flathead Basin. The lake trout were sampled all over, from Alaska to Michigan, all these different areas. And we said, well, why don't we use these this relatively large number of samples to try to identify diagnostic SNPs. But when we did that, when we were going to identify these diagnostic SNPs, we needed to exclude the quartz lake population because the quartz lake population was potentially hybridized. Okay? So what we did was we basically identified diagnostic SNPs. So the only samples we excluded were bull trout from Quartz Lake that, because that population looked like it may have been hybridized. And we identified about 19,000 diagnostic sites, so completely diagnostic SNPs between these two species. Okay? And the idea was if we look at these diagnostic SNPs and we apply them to these Quartz Lake populations, what proportion of the genome in these Quartz Lake bull trout looks like it is of lake trout origin. So basically when we did this experiment, here we had there's 20 Quartz Lake bull trout, 2 Quartz Lake trout, and what you can see is that most of the individuals are completely homozygous for bull trout alleles, but a few individuals, 2 individuals have basically about 25% exactly of their sites were heterozygous, so they had 1 bull trout allele and 1 lake trout allele, which is exactly what you'd expect if you had an F1 hybrid that back-crossed a bull trout 2 times, so 2-generation back-cross, okay? And so we had, 1 individual that was a little bit higher and some individuals that had a really low amount of lake trout alleles, so a relatively low proportion of their sites were heterozygous. But when we saw this, we thought, seems like there's some hybrid individuals in there. It's these 3 individuals again, but other individuals also have a lower seem to have a lower proportion of lake trout alleles, okay? So we're like, well, okay, we kept doing this. We thought these things really look to be hybridized. But one possibility we thought was, is it possible that because we excluded bull trout from Quartz Lake, that somehow that biased our diagnostic set? And then if we, so these weren't true diagnostic SNPs, but they appeared to be diagnostic because we excluded this one population, and these things really aren't hybrids. So what we did was we thought, well, let's come up with another diagnostic set of markers, and this time let's exclude Quartz Lake and Granite Creek. So we have a new set, so we excluded bull trout from Quartz Lake and Granite Creek. We identified diagnostic sites, so diagnostic alleles between these different populations. And the idea was, if we now look at Granite Creek bull trout, do they look like hybrids? Okay, so do we start to see things that look like hybrids when we use a different diagnostic set? So when we did this experiment, basically what we found was that Quartz Lake bull trout looked the exact same, so the same individuals look hybridized, and none of the Granite Lake bull trout appear to have any contribution, any any lake trout alleles in them. So this told us that it was not, we couldn't explain our result from possibly miscalling diagnostic sites. So it wasn't that we had somehow miscalled species diagnostic SNPs by excluding one population, and we actually repeated this with multiple diagnostic sets, so we excluded each of the different populations, called a new diagnostic set, applied the diagnostic set to quantify the proportion of ancestry in these Quartz Lake individuals and the other populations, and we never got any other bull trout from any other location to look like it had to have basically lake trout alleles in it. So, so at this point we were pretty much totally convinced. Like I said, we knew there wasn't contamination in the lab. We had gone through, we'd done this, we were like, wow, we've detected novel hybrids in Quartz Lake. And my student Zach was doing his master's, and he met with his committee, I remember, and his committee was saying, yeah, you guys published that, this is amazing. You got this connection to climate change and these novel hybrids, and as there's less and less snowpack, there's going to be more of them. It's this really amazing story. And so we thought, well, the only other possibility, so the only potential thing that we thought a reviewer might question was whether somehow contamination led to this pattern. Now, like I said, we knew that there wasn't contamination, because we had never had lake trout individuals in our lab when we prepared the bull trout samples, but we thought, well, why don't we go through and somehow formally try to exclude the possibility of contamination, such that when we submit it for publication, if any reviewer even brings that up, we'll have excluded it. So we thought to ourselves, well, how do we exclude the possibility of contamination? So does anybody have any idea how you could look for contamination? What sort of experiments you might do to test if these patterns were caused by contamination somehow?
Mix the DNA.
Yeah, so one of the things we did, I'll show you that in just a second, was we said, hey, well, why don't we take a pure bull trout sample and a pure lake trout sample, bioinformatically just mix the DNA at different frequencies, mix the sequence reads, and see what patterns we get. But how could you evaluate the difference between a 10% lake trout mixture and a 10%, and a real hybrid with, say, 10% of its genome coming from lake trout? Anybody have any idea? Yeah, you could potentially look for multiple alleles. So you could look for 3 alleles at one site, something like that. Multiple alleles at one site. It seems like a reasonable idea. Anybody else? So we were thinking about this, and this is what we thought of. We thought, well, if there's a true, so all these sites were heterozygous. They were called as being heterozygous. And so we thought, well, if there is truly, if sites are truly heterozygous, so they have one bull trout allele and one lake trout allele, when we sequence those sites, there should be equal representation of the 2 alleles. Relatively equal representation. Say we sequence 20 reads at one site, we'd expect approximately 10 of them to be bull trout allele, 10 to be lake trout allele. But if there's contamination, we'd expect where, say we had 10% lake trout allele contamination into our bull trout samples, even if a site was called heterozygous, we'd expect to have an allelic skew, where the lake trout alleles would be present at a lower frequency than the bull trout alleles, unless it was an exact 50-50 DNA mix, okay? So we said, well, why don't we look at that? And we came up with this expectation. So basically, if these individuals were really, 25% of their sites were heterozygous, so had one copy of a bull trout allele and one copy of a lake trout allele, basically we thought this is what we'd expect. So again, lab contamination was not possible, but we wanted to demonstrate that with this, what we call the diagnostic allele counts. So we sort of came up with this method. And basically what we did was we made a histogram where on the x-axis, it's the proportion of lake trout reads. So for every site, we have a particular number of reads, and some proportion of those reads are lake trout reads. So every diagnostic site we have that's heterozygous or that's homozygous. We have some proportion of lake trout reads, some proportion of bull trout reads. And so if we had a true individual that was 25%, so 25% of its sites were heterozygous, we would basically expect most of the sites to have 0, so 75% of the diagnostic sites to have 0 lake trout reads, and then the other 25% of sites to have approximately 50% lake trout reads and 50% bull trout reads. So this is what we reasoned. This is just made up. This isn't data or anything. This is just basically what we expected the distribution of allele count frequencies to look like in an individual that was truly a back cross, that was twice back cross to bull trout. Okay? So we said, okay, now that we've sort of come up with a model of what we expect that distribution to look like, let's look at our real data. And again, I'll sort of emphasize this. My student Zach was trying to finish up his master's. His committee was just like, graduate, publish that paper. What are you guys doing? We're like, well, we want to do some more tests and so on. And they're pressuring him to not to do this. We said, well, we want to come up with this method to check for contamination and this kind of thing. And so ultimately, we applied this test to the real samples, and these diagnostic allele count distributions of pure bull trout and pure lake trout basically conform to exactly what we'd expect. So when we took a pure bull trout, all of the sites had 0. So zero frequency of lake trout reeds in them. And when we looked at a lake trout, all the sites, they were called homozygous lake, and they all had 100% lake trout reeds, okay? But when we looked at one of these putative two-generation backcross hybrids, it didn't look anything like we'd expect. So this is what the data looked like, and this is what we would have expected. So the sites that got called heterozygous, even though they got called heterozygous, they actually had a much higher frequency of bull trout alleles than they did lake trout alleles. So these sites that were supposedly heterozygous didn't have 50-50 representation of the alleles like they should have if they were truly heterozygous. Okay? So we thought, well, I guess there's two possibilities here. Either this is contamination, but again, we knew it wasn't contamination. We had never had lake trout in the lab. That was our thought. We really hadn't ever had lake trout in the lab, actually. We thought, well, what if it's like an alignment issue? So what if somehow the lake trout reeds don't align as well to the bull trout reference or something like that, and some sort of alignment bias was causing a bias in the counts such that there was a higher counts of bull trout reeds at these so-called, heterozygous sites. So that's when we did what you suggested, is we made these artificial mixes. So we took pure bull trout, we took pure lake trout, and we mixed them at different frequencies. So we made a mix with 1% lake trout, a mix with 2% lake trout, 4% lake trout, 8% lake trout, and 50% lake trout. So these are, again, using just raw reeds from pure bull trout and pure lake trout and just artificially mixing them, right? So I don't remember exactly what we did, whether we used like head to grab 1 million reeds from one file and just concatenate them together with cat, whatever. We just simply mixed the reeds together. And what we found was that with different proportions of mixed reeds, we could actually recreate different proportions of heterozygous genotype calls. So for example, when we had 4% lake trout reeds, when we had a mix of 4% lake trout reeds and 96% bull trout reeds, we ended up calling that individual as having 25% heterozygous genotypes and 75% homozygous bull trout genotypes, which is basically exactly what a couple of those individuals from Quartz Lakes were, okay? So the question then was, what does the distribution of allele counts look like in these, in this artificial mix data? And basically, it looked very similar to our supposed bull trout hybrids, okay? So this is that 4% artificial mix, and what you see is that in the sites that were called heterozygous, they still had a very low proportion of lake trout alleles. They didn't have this equal number of lake trout alleles and bull trout alleles as we would have expected, okay? So at this point, basically, this convinced us that the only thing that could have caused this pattern was sample contamination. So it wasn't, these weren't hybrids. It was simply, we thought we discovered these new hybrids. We thought there was all this interesting biology, but it was actually just completely boring sample contamination. And so then the question was, well, how did the samples get contaminated, right? So Gordon sent us these samples, and so obviously, so I should have known right then what the problem was. I'm just joking. Basically, this is what happened. Out of all these sites, Quartz Lake was the only location where the bull trout were sampled in gill nets. So the bull trout samples that we got from Quartz Lake were just caught as bycatch when they were gill netting for lake trout. So when they're gill netting, they're pulling up these lake trout, like a whole bunch of lake trout. There's lake trout slime all over the boat, and every once in a while they pull up a bull trout and they take a fin clip off of it, but just lake trout is everywhere, right? So we had convinced ourselves that, it makes a lot of sense because this one location is the lake bull trout population, and Clint, had this anecdotal evidence, that they had pulled up these bull trout, and we were convinced, but in reality, it was just a complete artifact of the way that these samples were taken. And right now, it's very rare these days that the same person does everything, right? It's oftentimes the person who collects the samples is not who does the molecular biology, and the person who does the molecular biology is not who does the bioinformatic analysis, and all these things. And so the thought of field contamination had not crossed our mind at all. We didn't even think of that. And I think, I don't know, but based on the reaction of my students' committee, I think we probably could have got this through peer review and, published these novel hybrids between bull trout and lake trout, even though it was all just, completely artifactual, okay? And the other thing that's interesting about this is the only way we figured this out was we didn't use some sort of pre-existing methods. I think some methods are out there now to look for these kinds of things, but two things: we were skeptical, we were cautious, we were skeptical, and we just thought a little bit. We thought about an experiment we could do, basically. The experiment, we had to think, and we had to come up with an idea that would allow us to test the hypothesis that these data were created by contamination. And the basic idea had to do with the allele counts. The allele counts should have been 50-50, but they weren't. And so, just in general, you can talk all day about standard pipelines and standard analyses and doing all these things, but you always have to be skeptical of your data and just think about simple experiments you can do or simple patterns you can look at in your data to test, possible other explanations for the biology that you think you've discovered. Okay, is the sort of biggest thing for me. So this was a pretty interesting. So here's Quartz Lake. This is actually me and Sean. This was a trip with Gordon up to Quartz Lake. Sean is a project scientist in my lab. And here's them pulling up some lake trout, these are lake trout, and it's just kind of a mess all over the boat when they're actually doing this. So in retrospect, it makes perfect sense, and we should have known it was contamination, and we should have thought about it. But again, when that part of the project is out of your hands, and you just know what's in your control, I knew it for absolute sure. We had never had lake trout in the lab when we did those bull trout preps, so it could not have been lab contamination. But that doesn't mean it wasn't contamination. So that's just, I think a story that I like to tell, because I think it's very realistic. I think there's a relatively high probability that this type of thing could happen. It might not be contamination, but it could be any sort of artifact that you don't think about. Differences in Illumina changes their chemistry, and all of a sudden, there's new, or you use a new library prep, and all the structure you see is related to that, or it's related to lane effects. Anything you can imagine is possibly going on in your data, and you just have to be ready to be creative, be cautious, be skeptical, and think about, be cautious, be skeptical, and think about creative ways to try to prove yourself wrong, I think is the best way to think about it. So that's all I have to say. Does anybody have any questions? Yeah.
You got two.
Yep.
One, so I'm missing something. Why would you not ever, if these were contamination as opposed to hybrids, why
Yep.
would you ever pick up homozygous lake trout loci kind of thing as just overwhelming the bull trout?
Mm-hmm.
And two, could you not, when you're running your lake trout, if you ran lake trout, are you picking up the same kind of thing because of bull trout contamination?
Yeah, so, it looks like our contamination based on these artificial mixes that we made was at most about 5%, 5 to 10%. So it's very unlikely if you have 5 to 10% lake trout DNA, you have the whole genome coming in. You still have at any site, you still have mostly bull trout DNA. So you don't actually expect to have more lake trout reads. Just by chance, sometimes, on average, every site actually should have had about 10% or 5% or whatever bull trout reads and 5% lake trout reads. but just by chance you have a distribution. So some have a higher proportion of lake trout reeds and end up being called heterozygous, and others end up not having any lake trout reeds just by chance, because say you have 10x coverage, there's only 5% contamination. It's not unlikely that you won't sample a lake trout reed with 10x coverage if lake trout are only present at 5%, these kinds of things. Your second question was, do we ever see bull trout DNA in the lake trout samples? And no, we didn't. That was what was so convincing about this, is that just in this one single location did we see hybrids. We didn't see hybrids in any of the other samples anywhere else. So, and we had a couple lake trout samples from Quartz Lake, but the lake trout samples from Quartz Lake, especially early on when they started gill netting Quartz Lake originally, they were pulling up huge lake trout in massive numbers. And every once in a while you get a bull trout, So there's not enough bull trout present in the sampling to lead to significant contamination. And other locations, it's the same thing. The lake trout are basically, the bull trout are either extirpated or close to extirpated in many of these locations, is my understanding. So you just don't have significant contamination going the other direction. Any other questions? Yeah.
Do you think it's possible to have contamination between individuals if you pull them up from the same net and they're slime, they're the same species?
Oh yeah. So that's a great question. That's something that's really concerning to us now, is we realize how much contamination do we have all over the place, and usually we just don't see it. The reason we saw it here was because we looked, because it was such an extreme situation, but I would guess that there's been many papers, or lots of papers published, where they detected some admixture and these kinds of things. They ran structure, saw a little bit of what, and maybe attributed that, but it was just contamination. Or it's more difficult to look at it when you have similar populations. Jeff.
So you've probably thought of this, but so you can get a lot out of the distribution of the alternative alleles.
Yep.
And so this same pattern actually holds within a population. It's slightly different.
Yep.
So we used this. I've had contaminations within populations where there's no structure to flag it, like you had, where it was, there was no biological result, but it can cause kind of really a lot of screwy things with if you're doing set frequency spectrum or anything
Yeah.
with frequency. Even FST would be impacted by this. So if you polarize SNPs based on ancestral versus derived, and you can do this within any population sample, because most SNPs are rare, most contaminants actually systematically shift in one direction, and so any polarized set frequency spectrum over most population histories actually causes systematic shift into the derived allele frequency spectrum for individual heterozygous sites, and so you should always look at the distribution of your two alleles polarized in that group, and you can see a lot of, you can see cross-sample contamination, so between two bull trout, which might not be a management-worthy kind of contamination that would screw things up, but it's a really important check in data quality.
Yeah, so we actually wrote a likelihood function that would estimate contamination rate, and then it would incorporate that contamination rate into the likelihood calculation, but it hasn't been implemented or anything like that. But it's possible to rescue data that is contaminated. So basically you say, oh, well, if there's a 5% contamination rate, these sites, it's more likely that these are homozygous bull trout sites that just have a few reeds due to contamination, and if that's expressed in your likelihood function, then it can be corrected for, and you don't miscall. But again, we haven't implemented that yet. So there are easy ways to identify this. It's just not on your radar. You know what I mean? So it should be, in general. I can tell you to put contamination on your radar, and you should put that on your radar and think about it. But really, you should be cautious in general and be skeptical in general, because you don't know. We hadn't imagined that this was going to be causing an issue, but it ended up. It did cause an issue. So, yeah. Any other questions or comments? Yeah.
I'm just thinking about coverage, you know, like, let's just talk about one specific SNP, you know?
Yep.
And what X coverage, let's say that SNP, when you called it, I'm assuming you used like ANKS or something where you called that a heterozygote based on some type of Bayesian,
Yeah, just based on likelihoods.
right? So how many reads did you have at that location? And if you upped your reads, say from 5x to 20x, could you exclude that contamination?
Yeah, so in this situation, we actually have pretty high coverage. We have like 20x coverage. That doesn't change the, there is a skew. The math that was used to call these genotypes was basically exactly what we talked about yesterday morning. I think it was like a posterior of 0.95 or something like that. A uniform prior with a posterior of 0.95 for genotype calls, something like that. So upping the coverage would not solve this issue. But I will say this, if we would have had really low coverage data, I don't think we would have been able to test this contamination as easily. So we could easily see this because we had a lot of sites that had relatively high coverage and had a, 3 reads of lake trout and 17 reads of bull trout, that kind of thing. But even if you would have been more and more stringent here and required say you wouldn't have used the math that we talked about and used some, on top of that you would have used some equal allele cutoff or something. You still would have had some sites that were called as heterozygous, and these individuals would have shown up they would have shown up as 5% hybrids instead of 25% hybrids, something like that. Or have 5% of their sites heterozygous. So it's not there's not a really simple solution other than potentially using maximum likelihood to search for a contamination rate and then incorporating that contamination rate into your likelihood function. Or, you can look for it in various ways, and yeah. Any other questions?
I had a similar problem with 2 different species that were labelled as the same species,
Mm-hmm.
and found out much earlier on that in science sequencing. But do you think if you sent off these samples for science sequencing, like a barcoding marker, you would see like really messy sequence in your contaminated samples, and you'd know that they weren't good enough?
Yeah, so you would have seen like, your traces, a double peak, but one peak would have been really short or something like that. I don't really I haven't looked at enough Sanger sequence data to know if I would have seen that and said, it looks like it's heterozygous or it looks like a little bit of noise or not, so I can't really say. But yeah, there's certainly potential to discover contamination in various ways.
Microsats are excellent for finding
Yeah, so
contamination.
if you have microsats, you could see 3 alleles at a high frequency, or even 4 alleles, which would be a great way to do that. But again, I didn't tell you this for the purpose of contamination, to get you worried about contamination. I told you it more just to be skeptical of your data in general, because there's a lot of unforeseen biases that you just wouldn't have imagined, and the software isn't meant to deal with it for the most part, deal with unique things. So
that last phrase is worth re-emphasizing perhaps. It's important to have some scripting bioinformatics skills, because that kind of test or other tests that you might want to perform looking at your data aren't generally available. And
yeah, so this kind of thing, some of this, there is probably some software that does some of this right now, but at the time, we did this a few years ago. We didn't know of anything obvious, and I don't know if there was anything that was documented that could look at this at that time. But fortunately, my student Zach he just had some basic scripting skills, so he could easily parse just the raw files and look at the counts and build this distribution. So with just some basic scripting skills, instead of just like having an output file that you can plug into another output file. You can look at output files and you can easily look through tens of thousands or hundreds or even millions of lines and easily take that simple data, it's often just a tab delimited text file, and make basic plots like this. So just a little bit, you don't have to be a computer programmer and have a computer science degree, but having a little bit of capacity to just do some basic scripting allows you to just look at your data in a much deeper way. And so if that seems intimidating or overwhelming to you, basically the way I think of it is if you can open up a text file and you can just figure out what you want to do, like copy and paste these two numbers, divide it, and then do that 10 times and plot it in Excel or something. If you can do that, all you need to do is learn how to write a script to automate that process for you, across 100,000 sites or whatever. And so it's not, again, you don't have to be a computer scientist to just have some basic scripting skills to be able to just handle large amounts of data. So I would encourage people to try to develop those skills. It just made it so, in a week or a couple days even, Zach made these plots from the raw data, and we could look at them, and it wasn't an issue. But if you wouldn't have had that skill, it would have been much, much harder. Yes?
So with whole genome data, it seems like it's often in the pipeline to blast to remove contaminants. So, but with RAD data, I don't do a lot of analysis of RAD data, I just generate a lot of it, but from what I've read in papers, they don't often report the stuff like that. And so like if you're in the lab working, like extracting DNA, in our institution often we work on really tiny things. And I emphasize to students, don't, make sure you're just getting, for example, the little nudibranchs, make sure you're just getting nudibranch in that extraction, but some of them are really small, they might get some intestinal stuff in there. Is there a way to blast RAD data to
you can
identify that stuff?
Yeah, I mean,
Just remove it?
we've actually, it's pretty interesting. When you generate sequence, you never really know what you're going to get. We have, we were doing this mushroom project with some friends with albino, and we ended up detecting this, cryptic species of like 50 million years diverged, and the sequences, the RAD sequences didn't even align to each other from different samples that we thought were the same mushroom species. Remember we were doing that Paraperla project, Gordon, where a few of the samples, these insects, I don't know, was it 90% or something of the reads were actually viral in origin? So some of these bugs were just loaded with viral DNA, or the viral DNA had a high frequency of the particular cut site that we were using. Something like that. So there's a lot of different situations. In this situation, if we would have blasted, bull trout and lake trout are very similar genetically, and it wouldn't have been, but yeah, there's no reason you can't just take, so, the pipelines for doing that, that are applied to whole genome sequencing, there's no reason you can't apply them to your RAD FASTQ files, just like, so basically, yeah, you can do that. We often we don't do it by default, but we see patterns. Sometimes we'll see patterns and then we'll start looking, but it wouldn't hurt to do that by default, actually. Just grab the top 1,000 reads from your data set, blast them, and make sure they look like something that's related to what you think the data should be. Sometimes we've had libraries swapped by the sequencing center and stuff like that, so you'd hate to spend 6 months analyzing someone else's data, which I'm sure that's happened. Yeah.
So in the context of, we were talking about yesterday of de novo versus mapping, and so with the closed species contamination, that's a different issue, but some of the other things that come up are issues of a viral or species that are not going to map to your reference. I'm just wondering if you or other people have found with de novo sequencing, if that is more of an issue because you're not.
Yeah. If you have a reference for your species, you'll align it, and the other reads just won't align. I think that insect example that I mentioned, we did a de novo assembly, and we had a couple loci that were like the most abundant, best loci, and they were like a couple orders of magnitude more abundant in all the samples than anything else. And so we said, what's going on here? And we aligned them. I think we blasted them, and they blasted to something completely different, whereas the other loci blasted to insects and stuff like that. So yeah, if you have it's a good point, if you have a reference genome, and that's one of the reasons we don't just by default blast everything or whatever. But yeah, if stuff is in there, it's not an issue if it's really divergent, isn't going to line to your reference. There's always microbes like in everything. So just the frequency of reads from those will vary, obviously. Yeah.
So Gordon, how have you changed your field protocols?
I don't know, I didn't get in the field anymore.
Yeah, it was other people, and you know, at least
But in the lab,
we think it was the
if you just wash the fin clip thoroughly in the lab, no problem, Mike.
well, I'm attributing it to the field biologist, but there's probably like 10% chance it was people mispipetting in Gordon's lab, you know. So. I'm just joking. I think he sent us fin clips, not
Be
DNA.
careful to
So.
wash samples, I guess, of it.
Yeah.
Of whatever material.
Any
So,
other questions?
For my plant, it's an aquatic plant, and most of the reeds I get are not my plant.
Yeah,
The
it doesn't
majority are
surprise me.
The epiphytes and microbiome on the surface, so I just have to parse those out.
Back to the insect example, we had thought about, and I think we did use some abdomens and so gut material, and you know, there was an interest of possibly knowing what they're eating if we blasted red, you know, sequences, and we had, you know, information on what they're eating. Might learn something about diet along with genotyping the individual. And even diseases, there's a fish, tail rot bacterium that lives in the gut of one of these stoneflies we're studying. And there's, you know, it's possible, I don't know if anyone's looked into that, and then if you have a rat capture array and you target different sequences of interest, as long as you know what they are, you might actually get more information than, you know, sequences for more than the individual.
People are doing that for agricultural disease, things where they want to sequence the plant and the virus or the disease, so that's common.
You just have to be careful that if something's really abundant, all your sequencing effort, your reads might go to something else, like in the virus example, and not the individual you're hoping to genome.
Yeah, we've actually done some viral assembly out of salmon gill samples. So most of the reads come from the host, but you get enough that you can, especially these small genomes, you know, you just don't need many reads, and you can cover them really well. So, yeah.
Can that be more of a concern in, say, museum specimens where they might have higher bacterial content?
I'm sure it is. I would expect. The question was, is that more of a problem in museum samples? And I would certainly expect that.
And
to be the
depending
case.
upon the sample, there's cleaning protocols. So bone, you're supposed to wash really well before using it.
And that does the trick well enough to, supposedly.
Yeah.
Yeah, it really matters what the source of sampling is. Like with bone, the best way to do it is to drill in, because the outside has been in pins. You know, a lot of bones are deciphered from rocks by taste, the way they do in the field. It's horrible, right? Especially studying hominids. But if you drill in, you can usually get in away from that. Or like museum specimens, the contamination where it usually isn't a similar species, right? That contamination is really only an issue if you look, and you're careful, it's pretty easy to rule out distant contamination with bacteria by blast or something. So it's really only an issue with like things like Mike was talking about with the close related species that leads to biological interpretation. But with like the chipmunk stuff I talked about earlier today, and the bacterial contamination in those museum specimens is like 1% or something. It's pretty easy to remove with a standard blast pipeline. I think, Bryce, you have in that one cleaning pipeline that you have that includes bacterial contamination scan, right?
Yeah, so you could give it, well, actually, you just blast it for,
But it's part of that. Yeah, it's part of that.
yeah, but that stuff is just, you can identify any reads that any reference genome, so 5X, for example, is a big one in this course, or bacteria, E. coli is
everyone freaked out, Mike.
Yeah.
What's that?
You got everyone freaked out.
Yeah, you should be.
That's good.
You know, have fun, but be a little bit careful.
You gotta be
right?
extra careful. Any other questions about this topic in general? Being careful with your data analysis?
Maybe you could talk about some, oh, sorry.
Oh, I just have a comment. I heard this talk goes by.
Yeah.
We actually went back and redo type of conclusion
Oh.
based on this talk.
Huh.
And it worked. I mean, we had no contamination.
Okay. Sorry to cause the extra work.
Oh, I was gonna say, maybe we could talk about some other sort of common sources of bias or misinterpretation that pop up along these analysis pipelines. If
What do
anyone-
folks who do a lot of this think? Jeff, common sources of bias, misinterpretation?
I think one of the biggest issues, depends on what kind of analysis you're doing, but one of the biggest issues always for these approaches comes down to you comparing divergent populations or species is reference bias. If you have any step where you're doing either de novo assemblies and you're mapping to those or you're mapping to the genome, reference bias is a pretty big one.
Can you give an example of reference bias that you see, like what was the result of the reference bias? Like what,
What did it
you know,
cause?
so for example, I've aligned to different genomes and seen different rates, had different alignment rates. In some assemblies, I have more paralog issues, but I haven't had a misinterpretation of biology due to a reference. Have you guys had any issues like that?
Well, it's fundamentally flaws in the RNA sequence analysis where you have an F1 and you sort.
Yeah,
That's a
yeah.
big one.
Yeah.
That's a huge one. If you're doing so Bryce actually is working on some of this right now. If you're doing say a phylogenetic comparison where you're starting from one reference and you're either using RAD to go out a little ways or say capture to go out further or whatever. If you're always going back, you systematically will bias towards reference calls, which can look like hybridization across the phylogeny, and you also mess up branch lengths and all those types of things. So if you're any kind of phylogenetic inference, that reference bias can be pretty strong. And even with polymorphism levels or divergence levels that are on the order of 1% or 0.5%, you can get pretty reasonable, like say 5 or 10% reference bias can pop out. So it can be pretty strong pretty quickly, and if you went more and more divergent, losing rapidly evolving regions, dropping out haplotypes, things like that. Yeah.
You don't even have to get out of the species to have a problem like that. So I worked in the past a lot with mosquitoes, malaria transmitting mosquitoes, and one of the chromosomes, 40% of the chromosome is a chromosomal inversion, which is quite old. So because it's a chromosomal inversion, there is no recombination. And what we see is that the mosquitoes that are that have the chromosomal inversion, it's so diverged from the original format that the mapping that you get is much lower. So if you go there, it is the same species. You go to the reference and you map an individual that has an inversion. What you see is that you have less heterozygosity, but you actually don't have less heterozygosity. What you have is your mapping algorithm is failing. So you see how this area has lost diversity. And it's actually, after you correct for that, it actually happens that it's exactly the other way around. So the individuals that have the inversion, for some reason, actually do have more diversity than the ones that have. But it's a problem of mapping against the reference, and it is the exact same species. So you don't even need to go to a nearby species. You have a chromosomal inversion, they diverge, and then you have mapping problems.
So fewer reads mapped to that.
Fewer reads. You cannot relax the algorithm, because then the computational cost of the algorithm will take the edge of the universe. So that's normally not a possibility with CYP.
The mapping quality is used in most gene testing, right? So it's not just the pure reads map, but even if the same number of reads map, they're of lower quality. So you're not confident that as long as they're super biased. You can really bias a lot of these type of calls. In ways it can be pretty systematically strong. want to get away from all the reference bias. And so you can start with the reference and map to that, and then call genotypes, and insert those into the reference, and then iterate the mapping, and map again. And as you have a divergent region, you can actually iterate away from the divergent reference towards the species you're calling. And you can actually basically get a genome that is the genome of whatever it is with these islands of RADs or exons or whatever embedded in that. And so you have the spatial context, it's wrong, but it's an approximation of the spatial context. And then you can use that pseudo-reference as the basis to do whatever additional downstream mapping-based calling. So there's other tools that do that, but I think Redis is pretty easy to use. And it's available now. The paper will be out soon. But the tool's already available if you're interested in it. Maybe we can put that link on that. Send that.
I know a paper or two that has used RAD data for phylogeny and evaluated the effect of what you were discussing, branch length bias and making the topology change. So I can send that around. There were some serious problems that they were to describe. So I think it'd be worth briefly, so filtering, since Mike likes talking about it so much.
Gordon loves talking about filtering.
So how many people are doing non-paired and read RAD? And I would guess a lot of papers are being published. And I just want to loop back briefly to PCR duplicates. So, you can't remove them if you don't have paired and read. And we talked yesterday, I think, about PCR duplicate rates can be 30, 50, 90%. And that can cause allelic dropout or appearance of reduced heterozygosity. And it could happen in just some individuals. It might be some individual samples that are, maybe lower quality. It might be some individuals that are sampled in a different way. And so if you were to get, there's a number of problems that this could cause for your data and interpretation. But the scientific community, we don't know much about those problems yet. It would be nice if someone spent some time looking at the effects of PCR duplicates on genotyping error rates, if there's any sort of systematic effects. I remember in the days of microsats, chimpanzees, so allelic dropout was a big issue. It wasn't known in the early days of using microsatellites on low quality samples. And in a chimpanzee study I know, males were sampled from feces, females were sampled from hairs. There was allelic drop that was out in the males. Males looked within a group, more homozygous, more related. Papers published, cover of Science, suggesting males more related to each other. They don't differ so much as females. Pretty cool kinship inference, caused mostly by allelic dropout. And that sort of thing could happen due to PCR duplicates, I think.
If you don't use PCR in your RAD protocol, is that as big of an issue?
No.
Okay.
But
Yeah, it's not an issue at all. We should all be striving, if you're doing RAD, we should be trying to move towards PCR-free libraries. And for example, we haven't done it yet, but the new RAD protocol that we talked about yesterday that we're using is compatible with a PCR-free library kit. You just have to have a lot more starting DNA. In a lot of situations, you don't have that. You just don't have enough DNA to work. Jeff, have you guys used any PCR-free library kits?
Not for RAD or sure.
But for anything else though?
Yeah, we played around with them a little bit. They work really well. So John McCutcheon does, and he's in Montana as well, does a lot of PCR-free stuff. The best thing about PCR-free from the standpoint of sequencing genomes is it eliminates these inherent GC biases that are typical of most. Anytime you heat and cool DNA a lot, when you amplify it, you create systematic biases for the base content. And so PCR-free gets around that. I guess they work really well for some of those extreme genomes that are like really AT rich, for example. But I think it's a good idea. And if you're using something like double digest RAD or something like that, it's not just a symptom of like calling, you know, your genotype and you just want to remove it. Like that's one of the first things you can look at to see if your rad preps were good, right? The duplication rate and that variance. And that explains a lot of issues. And so like maybe some people in this room have had this issue with, you look at like double digest rad or single read traditional rad or whatever, where you're like, I don't understand it. I have dropout across all different individuals that appears random, but my coverage is high across all these different rad tags, but I just have a different subset of rads for every individual. But I don't understand how that could happen. Well, the answer is probably, I mean, you can't assess it with those types of data, but the answer is really easy, is that there was systematic bottlenecking of the complexity, like I talked about earlier today. So you just don't have very good representation, and so you're randomly dropping out subsets of the genome, you're randomly sampling, say, you know, this locus and this individual, because you only have a couple of copies of the genome in, you know, through whatever bottleneck step in all the manipulations, and then you amplify it, and you amplify right out of that. So you have high coverage, but it's basically like, you know, standard sequencing the same clone over and over again. Right? It doesn't tell you any, you know, it would drop sequencing error rate, is what it would do. But it doesn't, but it's hard to see that, because coverage is high, and weird dropout rates doesn't really make sense, and so using some kind of approach that allows you to evaluate what the complexity variance was across your libraries is really helpful philosophy there.
So I know it's an issue with metabarcoding that I'm guessing also non-metabarcoding studies that tag jumping, does anybody have experience with seeing that?
What is it, sorry?
Tag jumping and frequency that you've seen, but in literature I've seen pretty low reports of it, but I've also been to meetings where people are just freaking out about it, specifically EDNA meetings. So I wonder if anyone had experience with seeing that in the data.
I think you mean a common PCR in mixed libraries? Is that what you mean?
Yeah.
You mean swapping barcodes?
Yeah, swapping barcodes.
Yeah, it
You're
happens all the
pooling.
time.
Sorry?
It happens all the time.
All the time?
Yeah.
So not like
No, I mean, it
50%?
happens. No, it particularly happens anytime you have a really rare variant in the library relative to a common variant that'll be swapped out. This is why a lot of preps use dual barcoding, because it's much easier, because usually recombinant PCR happens from an incomplete amplification, and you pick it up on the next cycle and you complete it with the wrong, you know, the wrong bark, you basically get a mosaic, but that mismatches dual barcodes. So that's one of the reasons why that's used. So highly multiplex approaches should try and use, but in a meta sample is tricky because you're not individually in pooling, right?
Right.
So yeah, it happens. So one way to do that is to try and identify haploid markers that you can use and look for heterozygosity in haploid markers. So for example, barcode swapping in the mammal looks like heterozygosity in the mitochondrial tube. It's usually pretty easy to quantify the rate, and you can actually do a Denali estimate of it. That can help. Some level it's gonna always happen.
I mean, even if you don't have barcode swapping in your PCR, there's potential for, I don't know if you thought about this, the cluster, you know, there's not perfect assignment of read ones and read twos and read threes to the same fragment as there theoretically should be.
Yeah,
so no matter what you do, there's some low rate of misassigning reads to barcodes and misassigning fragments to individuals due to barcoding issues. And
so the really, like, the terrifying way to look at next generation sequencing data is that there's errors and problems everywhere. But the positive way to look at it is that that's been true of every single biological analysis you've ever done. But with next gen data, you can actually look at the distribution, model it, and control for it. You can figure it out. So that's the, this afternoon will last about silly.
Any other questions